Good morning, everybody. It's a great pleasure to have in-person meeting. This might be my third or fourth in-person meeting since the pandemic started, and the credit goes to the vaccines. Vaccines have saved more than 19.7 million lives, according to one of the reports at Lancet. But the challenge that we are facing currently is the emergence of escape variants. So, as you can see, this is the data from the Veteran Affairs Association showing that the efficacy of vaccine is reducing over time, mostly because of breakthrough infections. And this is not just limited to RNA viruses. In case of DNA viruses, for example, cytomegalovirus, a leading cause of birth defect and transplant failure, it has been observed, it has been observed in the case of renal transplant recipients that once a patient is put on valganciclovir, the therapy that is clinically very well appreciated, the patient starts showing emergence of like resistance for CMV, and when the virus was sequenced, it was observed that there were multiple ganciclovir resistant mutants, followed by which the patients were put on like foscarnet or letermovir, and unfortunately, the graft was rejected post-month 15, and the patient couldn't make it post-month 22, which makes one wonder. We are trying to design so many therapies. Is there a possibility for us to design therapies that could keep up with the evolutionary pressure of the virus? Which brings me to today's talk, antivirus with a high genetic barrier to the evolution of resistance. As I've already introduced myself, I'm Sonali Chaturvedi, a research investigator at Gladstone Institutes. And I'm going to talk about two new strategies that I worked on during my postdoc. And I hope I will be able to convince you by the end of my talk that these two strategies are escape resistant or have high genetic barrier to the evolution of resistance. So the first strategy that I'll talk about is therapeutic interfering particles, or TIPs. TIPs are defective viral genomes that compete with the virus replication and packaging machinery and transmits with the virus at R0 greater than 1. And while doing so, this therapy is going to learn from the virus and keep up with the virus evolution as it steals its machinery. And hence, we anticipated that this therapy or this strategy might be escape resistant. So I'm going to talk about TIPs first, followed by which I will introduce the concept of open loop lethality, which we just coined this year, and show that feedback disruptors, which disrupt autoregulatory feedback circuits in viruses for extremely conserved domains, lead to cytotoxicity. And the reason why we anticipated this strategy to be escape resistant is because to overcome the therapy, the virus would have to have mutation in the cis and the trans regulatory region of the virus to recapitulate the feedback circuit, which is an extremely rare occurrence. And this is just live cell imaging showing the intact circuit where the protein, the immediate early protein or the master regulatory proteins are autoregulated. And on the right-hand side, I'm showing you picture of misregulated protein circuit leading to apoptosis as I speak. So these are two strategies I'm going to talk about. Let's get started with TIPs, or therapeutic interfering particles. So for TIPs, first, I'll introduce the concept of TIPs, show its efficacy in SARS-CoV-2, and then we'll go over if TIPs can reduce transmission of SARS-CoV-2, because pandemic is majorly driven by transmission. So let's go back 80 years ago. A Danish virologist, Preben von Magnus, observed a phenomena in influenza virus where he observed this oscillation of virus titer over time, and he gave it a name as incomplete virus. When he looked at these virus particles, he observed that it had a lot of deletion mutations. So he observed this oscillation, which is also called predator-prey interaction. The concentration of predator increases to a point, it gets preyed on, and the oscillation keeps on going. Adeline Hackett, right across the Bay Bridge in Berkeley, observed the same phenomena in vesicular stomatis virus. VSV, as we all know, is a beautiful bullet-shaped virus. When she tried to recapitulate the same phenomena, she observed that there were stunted bullet-shaped viruses. Further, Alice Wang and David Baltimore repeated the same experiment and published this work in 1970s and coined the term DI particles. So even though we have these DI particles 50 years ago or 80 years ago, but their therapeutic potential hasn't been harnessed on. So let me walk you through what are DI particles and how we can attain the therapeutic potential from them. So defective interfering particles, by the definition, do conditionally replicate in the presence of infectious virus. So in the absence of infectious virus, they wouldn't replicate. They will only replicate in the presence of the virus. They do interfere with the virus replication. So as you can see, in the presence of a therapeutic interfering particle or a defective interfering particle, the virus titer would be lower, the infectious virus titer would be lower because the therapy is competing with the virus for its packaging and replication machinery. But what makes this defective interfering particle to have a therapeutic potential is having R naught greater than 1. So before the pandemic, nobody knew what R naught was. And this is a crowd of virologists, so we all know what R naught is, but just for like the junior trainees, R naught greater than 1 means one mother infection gives rise to more than one daughter infection. And that is the mathematical modeling conducted by my postdoc mentor in 2003 in JVI. And that laid rule to like how we can harness the property of these defective particles to have a therapeutic potential. At the beginning of pandemic, we hypothesized if we can create tips for SARS-CoV-2. We all know SARS-CoV-2 replicate and is pathogenic to the neighboring cells, transmits to neighboring cells and infect them. If we can design tips for SARS-CoV-2, these tip RNAs will compete with the virus's replication and packaging machinery, transmit with the virus at R naught greater than 1, and interfere with the infectious virus replication. So I'm not going to show you a lot of cell culture data, but we used the information that we had from the directed evolution as well as random deletion library sequencing, and I can go through them like in detail with you after the talk. But we identified 2 TIPS candidates, and we did a single administration intranasal dose of these TIPS using lipid nanoparticles in hamsters, in Syrian golden hamsters. And we used like 5 animals. We challenged them with 10⁶ PFUs of SARS-CoV-2, harvested lungs at day 5. And what we observed was the TIPS were able to reduce viral load by more than few logs. As these TIPS are part of viral genome, we wanted to make sure that as they conditionally replicate, we wanted to make sure if TIPS would induce any inflammation, because cytokine storm is the biggest challenge that patients suffering with SARS-CoV-2 face. To that end, we performed RNA-seq in these hamster lungs, and looked for any inflammation and observed that tips reduced inflammation consistently for all pro-inflammatory gene markers. And then we looked at the histopathology in the lungs and consistently observed that tips reduced the alveolar edema as well as infiltrates in the lungs that were treated with tips compared to the control animals, showing that tips reduce viral load, inflammation, and pathogenesis. We have also looked at the R naught and transmission of tips, and we were not able to see the transmission of tips very efficiently in animals, even though we were able to see it in cell culture. So going to the part 2 of this work, so after we were able to show that tips reduced inflammation and virus replication in hamsters, we wanted to see if tips would reduce transmission of SARS-CoV-2 in hamsters. And the reason why we care so deeply about this problem is, for example, both my parents have pre-existing conditions and they are predisposed to COVID. And I have friends who are immunocompromised. So the patients for whom vaccines wouldn't work very efficiently because of the immunocompromised status, we can reduce the household transmission of virus if somebody can take tips and reduces the transmission to unvaccinated population. To that end, this is just a hypothesis that we worked on when we started working on this project. So in case of wild-type replication of SARS-CoV-2, we know that hamsters shed the virus. And that's why most of us are masked here, because humans shed virus a lot. And we hypothesized that as this is a single intranasal administration therapy, and the first site of replication of the virus as well is nose, we anticipated if we can start with we provide single administration to hamsters and see if that can reduce the transmission. And to that end, we performed a bit complicated experiment. And this is the time when Delta was a very big problem. Omicron had just shown up. And apart from being very transmissible, we all have seen that Delta is extremely pathogenic. So the previous experiment that I showed you was a prophylactic treatment of tips, where we administered the therapy and then treated the patient, or the hamsters in this case. In this case, we infected hamsters at a high titer of the Delta variant, followed by which we treated hamsters with either tip or control LNPs. We co-caged animals. So the animals that were infected and treated are golden ones and will be called as source animals. The animals that were naive, that is neither treated or nor infected, are called contact animals and are silver animals. After co-caging for 8 hours or partying, these animals were quarantined individually in cages, and we harvested their nasal washes and got the lungs at the end point and looked at the virus titer in the mucosa of these animals. We observed that in case of source animals, tips treated animals consistently showed reduction in the virus titer as anticipated, because that's what we saw in the previous study. But what was very exciting for us was to observe that the contact of tip-treated animals also showed or also demonstrated reduction in virus titer. And the reason why you're seeing this decline in the virus titer even for control and tips-treated animals is because after 6 days of infections, hamster clear off the infection. So the virus titer reduces to day 6. and the virus becomes undetectable post that. But consistently, even for contact animals, we observe reduction in the virus titer. When we looked at the lungs, we observed that the virus titer was lower for source and the contact of source animals treated with tips, showing that tips reduced transmission of SARS-CoV-2 in these hamsters. Further, we looked at disease pathogenesis in hamsters and observed that tips consistently reduce severe alveolar edema and infiltrates in source as well as contact animals. And when we looked at composite histopathology, we observed that contact of tip-treated animals are way healthier. To summarize this part of the story, I showed you that tips downregulate SARS-CoV-2 replication for multiple variants. I showed you that tips reduce transmission of SARS-CoV-2 in contact animals. And then I showed you that tips reduce disease pathogenesis in source and contact animals. Which brings us to the acknowledgement part of this part of the talk. I want to give a huge shout out to Lior Weinberger, my postdoc mentor. He has been working on the concept of tips for the past 2 decades. And this is the first tips that got published, so it's all because of his perseverance. Davey Smith and Tom Rogers at UCSD and Scripps, Robert Roddick, who is our partner in crime. He is CEO of VX Biosciences, the startup that Lior co-founded. And he's taking this therapy to clinic. And Leanne Minell, who has worked in the Pfizer vaccine development team, and she's helping us with the LNP production and all. Now let's go to the second part of the talk, which is feedback disruptors. This is the outline of my part 2. I'll first introduce why do we care about cytomegalovirus? Come on, we have so many RNA viruses to work on. Then I'll talk about why transcriptional feedback circuits. I'll walk you through the strategies we use, and I hope I'll be able to convince you that this is an escape-resistant therapy. Let's get started with the unmet clinical need of cytomegalovirus. So CMV is the leading cause of birth defects in the United States and worldwide. So this is an image of a patient or a child suffering from severe case of CMV. And you can see a lot of cysts in the CNS region of the child. CMV is also an opportunistic pathogen and is usually causes secondary infection and causes retinitis, especially in the patients suffering from HIV. And more than 60% of solid organ transplant recipients are infected with CMV, making it a very important virus to work. But the challenge the field of antiviral therapies is facing in case of CMV is major dose-limiting toxicity and resistance associated. It's a large genome. It is close to 230 kilobase pair in size, and this virus very beautifully expresses its gene in a cascade. That is, it expresses immediate early genes that in turn activate early gene, which in turn activate late genes. So the gene that I targeted was immediate early genes, or IE gene. So let's go and look at what these IE genes do. So once a virus infects cell, the DNA enters the nucleus. It expresses this immediate early gene, which transactivates downstream gene expression. While doing so, it autoregulates its own expression. And while we were working on solving the structure of this protein DNA complexes, we hypothesized if we can disrupt this autoregulatory feedback circuit by a competitor, a feedback disruptor. It doesn't have to be oligonucleotide, but in this case, we use double-stranded oligonucleotide, which are going to compete for this immediate early protein for the cis regulatory element present on the promoter. And By doing so, they are going to disrupt this autoregulatory feedback circuit, which in turn would induce cytotoxicity. And the reason why we anticipate this may lead to an escape-resistant therapy is because to recapitulate the feedback circuit, there have to be a complementary mutation in the protein and the cis-regulatory element. And it is an extremely rare occurrence. So this was the theory, why we cared about this problem, and like how we tried making a therapy that may be escape resistant. I'll walk you through the approaches that we developed. So first we did biochemical approaches. We did a lot of binding assays, like a true biochemist. We looked at the oligomerization of protein under EM, find a sequence that binds most efficiently with the protein, and tested it in cell culture. And this is a minimal feedback circuit, cell line, which just has the promoter, the protein of interest, that is immediate early protein, and GFP as a reporter. And it was predicted if there is a feedback disruptor, we would anticipate overexpression of GFP, leading to cytotoxicity. On the right-hand side, I'm showing you flow plots of either scramble sequence of feedback disruptor treated sequences. And the feedback disruptor treated sequences led to overexpression of GFP. And on the right-hand side, I'm showing you the cytotoxicity associated with this therapy. At the bottom, I'm showing the statistical significance and quantification of percentage GFP and dead cells, showing that CRS disrupts I86 negative feedback circuit in a minimal circuit cell line. We performed extensive RNA-seq experiments and looked for the fold enrichment of the genes, the host genes, and observed that the most enriched gene was extrinsic apoptotic pathway, showing that FDs induce apoptosis in a minimal feedback circuit. And then we asked ourselves, what would these FDs do in an infection setting, in a virus setting? To that end, we infected cells that were treated with FDs or scramble with cytomegalovirus and looked for the expression of immediate early genes and observed that at early time point of infection, the I86 gene was overexpressed in FD-treated samples. And we observed that at multiple MOIs, the FDs were able to reduce virus titer efficiently compared to scramble or mock infection. We took this thing and we wanted to see what would happen in a systemic infection in mice. So we designed a FD for MCMV or murine CMV and treated either at 3 hours pre-infection, treated at 3-hour pre-infection and 18-hour post-infection of MCMV, and harvested liver and spleen at 5-day post-infection. And what we observed was, when we looked at the MCMV mRNA, we observed that I88 protein, which is 88 kilodalton for MCMV, was overexpressed, whereas when we looked at other viral proteins, they were downregulated in FD-treated samples, showing that there is overexpression of I88. When we looked at the DNA, the viral genome, we observed that FD88-treated animals showed reduction in the viral genome. When we titered the virus, we observed that the virus titer was significantly reduced in liver and spleen of FD-treated animals. And when we looked at the percentage initial weight, we observed that FD-treated animals had gained weight, whereas the animals that were treated with the vehicle led to reduction in the weight of animal as anticipated. Now, fine, I designed a therapy, it's working, so what? Because we don't just want to make therapies, we want to make therapies that could have a high genetic barrier to evolution of virus. To that end, I did this experiment, which was an extended culture experiment. Yeah, I did that. So we, sorry. So we infected FD-treated or mark-treated cells with, with cytomegalovirus. Either we titered that sample, at the same time we transferred it multiple times to the cells that were treated with FDs for 60 plus days. And we titered all that virus, and we used formiviricin, which was the first antisense oligonucleotide to be ever FDA approved as a negative control, because formiviricin leads to resistance of the virus very quickly. And we observed that FD-treated animals consistently reduce virus titer, and after day 48, the virus goes down to detection limit. For HSV1 as well, we designed FDs and showed that FD-treated samples reduce virus titer below detection limit, showing that it's an escape-resistant therapy. To summarize this part of the talk, I showed you that FD breaks negative feedback circuit in cell lines, in minimal circuit cell line. It reduces virus replication by more than 100-fold in culture and in mice, and it's an escape-resistant therapy. To acknowledge this part of the talk, I want to give a huge shout out to Lior again, who is incredible support, and the feedback, the minimal circuit cell line that I showed was what he made during his postdoc, and all the other co-authors on this work. But I want to dedicate this part of the talk to Cynthia, who was my mentor and played a very crucial role in my herpes virology training. And so I showed you 2 new therapies or new strategies to design escape-resistant therapies. And with that, I'm ready to take any questions that you may have.
Folks with questions, can you please approach the microphones? And we have about 4 minutes for questions.
That was great. So much really awesome work. I have a question about your tips. Do you know if the tips actually get transmitted to the bystander hamsters, and could that be?
Yes, so that's a great question. And so I did mention that in cell culture we do see transmission, but in hamsters we are not seeing any significant transmission. And we are working with FDA, and they are very conservative on the transmission of tips, but we can make tips more transmissible or less transmissible if required. But for now, no, we are not seeing any significant transmission.
But you're looking early too, right? Because you did your checks on day 6, so you probably at that point you wouldn't expect to see them anymore. But maybe day 1 or day 2.
Maybe. But then we do see the virus by day 5. So if tips is getting transmitted, we would anticipate to see it as well. Thank you for the question.
Hi.
There's one in the back first there.
That's okay. Lisa Purcell from Vir. So
Hi.
Great talk. Can you comment a little bit on the variability in your tips? So when you look at viral load, you're seeing probably 20%, maybe a little bit more, that don't have differences in actually replicating virus. And so can you talk a little bit about that and what the implications might be?
So we have observed consistently there is one animal that doesn't show efficacy. And when we looked at the amount of M-cherry which we used as a reporter for tips, we observed that that animal didn't take the tips very well. So maybe they did sneeze tips because it's an intranasal therapy. So we did struggle a bit with that. So we are anticipating it is because of that reason. Yeah, so for humans, we are designing devices, but with hamsters, there is always one outlier.
Hi, Steve Mason from Janssen.
Hi.
I was wondering about the tips. What kind of dose do you need to use, and what is the time that they stick around intranasally?
Yeah, so with tips, we are using close to 100 microgram of RNA per hamster. It ranges from 60 to 100 microgram. We have done tip treatment 12 hours post-infection, 12 hours pre-infection, and 6-hour post-infection as well. But we are looking into how early you can give a tip and still see efficacy. So we are conducting those experiments. Thank you for the question.
Hi, John Malello from Gilead. Kind of a similar question, but since the tips replicate with the infectious virus, I was wondering, and appreciating how difficult the experiments are, if you've titrated out the level of tip per virus that is effective.
We can definitely do that. We haven't done that yet, but that's a great suggestion.
We have time for one more question.
Yes, please.
Okay, this is, first of all, there's a couple parts. How frequently does, since it replicates exuberantly, how frequently does coronavirus actually generate its own defective interfering particles? And is it possible in your system, there actually is a known strategy for inhibiting resistance, and that's to use multiple drugs that target different targets. Can you actually incorporate another antiviral component like a CRISPR system into your TIP, and have you tried it with other drugs to see if you can further limit the resistance? And I just have one quick comment. In your slide, I saw Arjun Kumar, who actually got his first job out of college as a result of attending this conference. So that was actually fun to see.
Actually, I also got my job while watching YouTube video from this conference. So thank you for all the organizers. I got my postdoc job because I saw Leo's talk at BayViro.
Okay.
So thank you for the question. I'll be very quick. Yes, there are naturally occurring defective interfering particles, but we are engineering them to make them more transmissible at R naught greater than 1. There are a few reports. There is a very recent report by McGill University where they have identified defective particles. The second question was if we can use a CRISPR technology or other approaches. The only challenge with that, we can try that, but it would be interesting to try. But we are trying to make a therapy that evolves with the virus, so we are not sure if like the other strategies would be able to keep up with it. But we can use we have designed FD, a feedback disruptor for SARS-CoV-2, and we can try using both together and see how these therapies can work synergistically. Thank you for your question.
Thank you.